Fantastic. So it is my privilege to introduce Clayton Agler for his senior experience talk. Clayton comes to us from the great state of Ohio and really approached me earlier in the year to think about giving a talk on medicinal chemistry. And then when I started drawing a bunch of hexagons, we had to slow down and say like, well, let's think a little bit more broadly about medicinal chemistry. And I pushed Clayton to really go read about how these drugs might be working for both malaria, schistosomiasis, and other compounds, and to get to a point where he could teach me how they work. He got to that point shockingly fast, which says something good about me, or good about him, or bad about me, but I think maybe both. And so the work that he's going to present today is an outgrowth of that. So a pretty deep reading and understanding of literature, and then implementing that in the lab, taking apart mice, pulling out parasites, very exciting work. And I'll let Clayton take it from here.
All right, thanks for the introduction, Stefan. I'm going to share my screen here. And as Stefan said, my talk today is going to be about blood-sucking bugs, or these different parasitic diseases that digest hemoglobin, and the war that these parasites have to fight between oxidative stress and hemozoin crystals. Obviously, today there's going to be an emphasis on medicinal chemistry and drug design, drug mechanisms, things like that. Also with some public health things sprinkled in there as well for some broader application. A few things that I'd like to point out here at the beginning. First, I have these slide numbers down here in the bottom right-hand corner. If anyone has any questions, at the end of the talk, there'll be plenty of time for questions. And if you just want to jot down the slide number that you have a question on or anything like that, you're welcome to do so. And also I have this blue box up here in the right-hand corner that you can use Zoom to put my face in if you so choose. And then I'll be part of the PowerPoint and part of your talk. So without further ado, we'll get going here. The first disease that I'm going to talk about is schistosomiasis. Schistosomiasis is prevalent in Africa and many regions throughout the world. Here we have a map where you can see the main areas that schistosomiasis is prevalent. You can see here Africa, and primarily sub-Saharan Africa, is where the most cases of schistosomiasis are. We also have plenty of cases in Brazil and a few other South American countries, and also some Southeast Asia countries, as well as the Middle East has some cases. So this is a disease that affects a large portion of the world's population. There's a lot of people affected by this disease every year. You can see over there to the right, there's about 252 million cases per year, and those cause about 200,000 deaths per year. And then I also put the mortality rate there. The mortality rate for these diseases is pretty low. The main way that these diseases affect and really hurt humans is by production of the daily household and people constantly being sick and things like that, as well as obviously a good amount of deaths. I'll quickly go over the life cycle of schistosomiasis. The life cycle has three main stages. 2 stages occur in the human and 1 stage occurs in the snail. So I'll start down here in the bottom right-hand corner, as you can see the laser pointer here. The life cycle starts out with these eggs, and these eggs mature into Miracidia. The Miracidia is the form of the parasite that infects snails. And these snails live in these freshwater bodies of water. So in these countries, again, this disease mainly exists in sub-Saharan Africa. And these freshwater bodies of water are home to where these people do a lot of their daily activities. They wash, they bathe, they do cooking and cleaning in these bodies of water. And these are just communal bodies of water where they rely on to do a lot of their everyday activities. And this is where the disease and the parasite, the Schistosoma mansoni, exists. So again, these Miracidia infect snails. And then inside the snail, the Miracidia mature into these Cercariae. And the Cercariae is the form of the parasite that is infectious to humans. These are microscopic organisms, and they can penetrate the skin of humans. They essentially just pierce the skin, and it's generally not something that you can even feel or hurts or anything like that. They just enter the body that way, and they can enter the blood vessel. And then once they get in the blood vessel, they kind of have a tour of the human body, and then they end up migrating around the liver. So as you can see up here, a lot of the parasites end up in the liver or in the blood vessels surrounding the liver, and then they can mature into adults. And we see here the adult forms of the parasite. The male and the female worms actually look fairly different. They actually look like two different species. And you can see here the female is generally inside the male, or the male is kind of wrapped around the female. And some research call this like the cuddling position, or with the male being the big spoon and the female being a little spoon. And that's an easy way to think of it. And these adult worms are about a centimeter in length or a little shorter, depending on the type of organism that they're in and the host and things like that. And then once these adult parasites develop, they can reproduce and produce more eggs. And then these eggs are excreted by humans back into these bodies of water. And that's how the life cycle of the parasite can continue on. And now we're going to talk a little bit about malaria. Schistosomiasis and malaria are the two diseases that I chose to focus today's talk on. But I would like to call out that there are a lot of parasitic diseases that this talk is applicable to. There's a lot of parasitic diseases that have hemoglobin digestion somewhere in the life cycle. Otherwise, they feed off of the human blood in some way to exist. And a lot of the concepts that I'll talk about today are common to all those parasites, even though I'll only focus on malaria and schistosomiasis today. So again, malaria, very similar to schistosomiasis, affects the same areas of the globe, mainly sub-Saharan Africa. But again, we have some more cases in Southeast Asia over here, South America, and the Middle East. Very similar case numbers, 229 million cases a year, about 400,000 deaths, with again a low mortality rate. But again, malaria causes this burden on households that impacts the production of households and causes people to be sick a lot, and also this good amount of deaths that it causes. And now we'll go through the malaria life cycle quickly. It's very similar to the schistosomiasis life cycle, so I won't spend quite as much time on it. The mosquito here is essentially the snail of malaria, if we're comparing it to schistosomiasis. It's the other non-human host that's required for the life cycle to continue. So the infected mosquito, when it bites the human or sucks the human's blood, or however we want to look at it, these sporozoites from the mosquito go into the human bloodstream and they settle in the liver, very similar to schistosomiasis. And then in the liver, they mature into these mirazoites, and the mirazoites are released into the bloodstream and infect erythrocytes, or red blood cells. And in the red blood cells is really where the disease progresses in humans and where the symptoms can occur. And essentially the way that it attacks the red blood cells is very similar to the way viruses work, where one of these mirazoites will infect a red blood cell, and then inside that red blood cell, it'll multiply and replicate, and then eventually lyse the red blood cell. And then all of those 20 or more mirazoites will go infect new red blood cells. And that's how the disease can spread in humans. So quickly, I'd like to define oxidative stress and its implications in the human body and in organisms in general. Oxidative stress is caused by molecules with redox potential, like this iron 2 plus here that I have. Iron 2 plus will be very important in today's talk, and is the main way that oxidative stress is carried out in all these organisms. So oxidative stress causes apoptosis, or programmed cell death, and it causes this in the parasite. This pathway is common to all living organisms, but again, these parasites are living organisms, and oxidative stress can lead to killing the parasite, and that's the way that a lot of these drugs work. So to create a simple analogy, parasites digesting hemoglobin is similar to the way we digest Taco Bell. So Taco Bell, a lot of us eat Taco Bell. Taco Bell tastes good, gives us nutrition. things like that. And in a very similar way, the parasite digests hemoglobin. Hemoglobin is essentially the parasite's food. So again, when we eat Taco Bell, we get nutrition, we get joy, and the parasite gets amino acids. And these amino acids are essential to the parasite. It's what allows them to replicate and live and survive and create more offspring. But we also get problems sometimes when we eat Taco Bell. One problem that we can get is indigestion. Sometimes Taco Bell doesn't make us feel very good about an hour after we eat. Well, these parasites have a similar problem that when they eat this hemoglobin, it produces heme. And heme is a bad thing in the cell. It causes that oxidative stress that I just talked about. So we need a solution to this problem. The solution for us when we eat Taco Bell, sometimes we take a little Pepto-Bismol or some antacid tablets or whatever makes us feel a little bit better. The parasite forms this heme into hemoglobin, or into hemozoin, sorry. So this heme is a bad problem for the cell. The heme causes a lot of oxidative stress. And to solve this problem, it takes that heme and forms it into hemozoin. So to create a simple schematic, we can think of hemoglobin as this box with a bunch of different shapes in it. Hemoglobin is a complex molecule. It's a pretty big molecule, and it has a lot of different parts and pieces. But we're going to focus on the circles here. These circles are what I've made up to be heme. So here we can say heme is just a circle. So heme is just a part of this bigger molecule that's hemoglobin. And then hemozoin is just a lot of these heme molecules essentially globbed together. So we go kind of from a bigger macro scale of this big molecule of hemoglobin to a small subsection of it, which is heme. And then again, hemozoin is just a polymer, a big glob of these heme molecules. So here is a more realistic schematic of what this actually looks like in a red blood cell. Here we can see a red blood cell. And then inside the red blood cell, we have this digestive vacuole. And right here, this digestive vacuole is where I want to focus our attention. So again, the parasite infects this red blood cell. And then once it gets in the red blood cell, it goes into this digestive vacuole. And inside the digestive vacuole is where all the magic happens. So it takes this hemoglobin, just like I showed before on the previous slide, and it digests it into peptides and these amino acids that the parasite needs to replicate and survive. It also makes this heme when it digests this hemoglobin. And then the heme again causes problems for the cell and it forms it into hemozoin. So again, heme is a dangerous problem for the cell. Hemozoin is the solution to that problem. And this all happens in these digestive vacuoles inside the red blood cell. So here's a molecule of heme. I'll just quickly walk through the molecule of heme because it's pretty important and central to my talk. We have these aromatic rings that are kind of all around the iron core here in the middle, and these nitrogen atoms coordinate the iron core. And then the other important part of the molecule is these two carboxylic acid tails that it has coming off on each side here. Here we can see a molecule of hemozoin. So again, hemozoin is just a big polymer of a bunch of individual heme molecules. So here up here, you can see this is a heme molecule, just like I showed in the previous slide. This red part here in the middle is the iron core, and we have these blue nitrogens surrounding it. And then the aromatic rings and these carboxylic acid tails. So these carboxylic acid tails is how the hemozoin polymer can be formed. So essentially, this is a long chain of dimers of heme molecules is one way to look at it. So here we have like an individual dimer. So in this dimer, we have one of the carboxylic acid tails from this top molecule coordinating the iron core below it. And then likewise, we have this iron core over here being coordinated by the carboxylic acid tail of the opposite heme molecule. And this creates this dimer. And then since each molecule is only using one of its carboxylic acid tails to create the dimer, the other tail is used to attach to another dimer. So this is how all of these dimers can be connected here. These are hydrogen bonds between the OH and the O, respectively, of each carboxylic acid tail. And that's how it can create a big molecule of hemozoin. So that's the the nuts and bolts chemistry of how heme is converted into hemozoin. And another important note here, this polymerization of hemozoin occurs under slightly acidic conditions. So like between pH 4 and 5. And that'll come up a little bit later in the talk. So now we'll get into a little bit of the drug development of how past drugs have worked and how new drugs could potentially work. The industry standard drug for treating malaria right now is chloroquine. There's recently been a lot of resistance against chloroquine in countries all over the world, especially in Africa. There's a lot of chloroquine resistant forms of malaria now. So here in the bottom left-hand corner, we can see a molecule of chloroquine. It's just got these two aromatic rings here with some nitrogens, as you can see right here. And then we have a molecule, a heme-like molecule up here. This isn't exactly heme because it has a slightly different core, but it behaves the same way as heme does. And you can see over here on the right how chloroquine binds to heme and prevents the formation of hemozoin. So you can see over here that this oxygen here coordinates with what would be the iron core of a heme molecule. And this essentially goes on top and on the bottom of each dimer. And that way, these dimers can't attach themselves to other dimers with these carboxylic acid tails. This carboxylic acid tail here gets kind of covered in a way, and so does this one at the bottom. And then heme molecules can't come on top and bind to the chloroquine. So chloroquine doesn't work by ripping apart hemozoin polymers, but rather it just prevents the formation of bigger hemozoin molecules and causes a slight buildup of heme in the cell, which causes oxidative stress. So here I just have a simple schematic of how chloroquine works. We have the original schematic here that parasites that essentially eat hemoglobin, which gives them heme and amino acids. And then they form this heme into hemozoin to solve the problem of oxidative stress. Chloroquine comes in during this area right here and prevents the formation of further hemozoin, and that leads to parasite death via oxidative stress. So here is a figure from one of the papers that looks at the mechanism of chloroquine in the malaria disease. So here on the y-axis, we have parasite survival on the left and free heme on the right. So this free heme is the amount of heme in the cell that's not formed into hemozoin. So as you can see here, like at about the 10 mark, this would be about 90% hemozoin and only 10% free heme. And then on the bottom here, we just have an increasing dosage from left to right. And as you can see here, as the dose increases, the parasite survival percentage decreases, which we would expect. The more drugs we give the parasite, we would hope that more of them die. And then on the right here, we see that the free heme increases as the dose increases. So again, this is hopefully what we would expect. We know that chloroquine causes a buildup of free heme, so we hope that the more chloroquine we give a cell, the more free heme we see in that cell. And then this red line here in the middle denotes like the IC50 or the EC50, the effective concentration to kill 50% of the parasites, which is an important number in drug development. And we can see that to kill about 50% of the parasites, we only have to have about 10% of free heme in the cell. So again, we don't have to cause all of the heme in the cell to be free heme, and we don't have to completely prevent the formation of hemozoin. We actually just have to slow it down. And if we slow it down enough to even about 10% in the cell, we can kill a lot of the parasites that way. So this got me thinking that, again, a lot of research has been done into chloroquine and malaria and things like that. And a lot of this research has to do with, we want to prevent the formation of hemozoin to cause this buildup of oxidative stress in the cell. And that way we can essentially kill the parasite through this oxidative stress. Well, malaria isn't the only parasitic disease that has this common pathway of the parasite digests hemoglobin to produce heme and then forms hemozoin. There's a lot of parasitic diseases that do this. And again, one of them is schistosomiasis. The life cycles are, again, very similar. They feed off the blood of humans, and that's how they proliferate. So I thought to myself, well, if chloroquine works in malaria, chloroquine should also work pretty well against schistosomiasis and preventing the formation of hemozoin there and causing the buildup of oxidative stress. So that's what my research here at Lawrence looked into, is attacking this common pathway of the digestion of hemoglobin and seeing if we can produce drugs to target that pathway. And it's important to note that this isn't the only way to kill these parasites. There's tons of ways to kill these parasites. This is just the avenue that I chose to go on. And here is a good example of that. Praziquantel is currently the industry standard drug for treating schistosomiasis. And there's actually not really any resistance to praziquantel, which is a problem that we have with malaria, but not right now with schistosomiasis. So praziquantel is this molecule we can see here. And again, it doesn't look a whole lot like chloroquine. A lot of the features of the molecule are pretty different, and it doesn't affect the formation of hemozoin at all. It actually attacks a completely different pathway. The mechanism for how this drug works isn't completely agreed upon by scientists, but it's generally agreed upon that praziquantel attacks the nervous system of drugs, and it causes it attacks the nervous system of the worms rather, and causes the worms to shrivel up and essentially be paralyzed. And it doesn't necessarily kill the worms, it just makes them shrivel up and detach from our blood vessels so that our body is able to flush them out. And that's how the drug works. So here we can see a study that actually treated schistosomiasis with chloroquine. So this was my original idea, to just treat schistosomiasis with chloroquine, since the two parasitic diseases share this common pathway. And there was actually already a study that did this. So we can see here on the y-axis, we have the number of worms per mouse. And on the x-axis, we have the different control groups and the chloroquine groups for males and females. And we can see that when they treated the worms with chloroquine, they saw about a 50% reduction in the amount of worms per mouse. So again, they essentially just treated the worms with the drug and then did an autopsy on the worms to see how many worms there were in each mouse. And there was about half the amount of worms in the chloroquine mice compared to the control mice. And it is worth noting that although this difference is significant, if they were to treat praziquantel, we would likely see 0 worms per mouse for praziquantel, or close to 0 worms, because praziquantel works a lot better than chloroquine at treating schistosomiasis, which again is why chloroquine isn't used to treat schistosomiasis. But again, this common pathway is worth targeting and worth more research into. So my research here at Lawrence involved taking these 3 compounds. These are novel drug substances that Stefan Debrett actually created in his lab here at Lawrence. And they were tested by the Kaiser lab in Switzerland against these live schistosome worms to see how these novel drug compounds did at killing the parasites. So the Kaiser lab in Switzerland found that for the adult worms at these 2 doses that are pretty small doses, after 72 hours, for the 1 micromolar dose, about 90% of the worms were killed among the 3 compounds that I tested here at Lawrence that are highlighted here in yellow. So these 3 drugs worked pretty well in this Kaiser lab in Switzerland and killed a good amount of the worms. So that's why I'm looking into these 3 compounds specifically. And the 2 research experiments that I did here at Lawrence for my senior experience was first trying to replicate the results of the Kaiser lab in Switzerland. We didn't use their methods. We actually used a different paper's methods to test the viability of these schistosome worms against these drug compounds. The assay that we used is very similar to the one that the Kaiser lab used, but different in some ways, just to see if we could replicate those results in a different way. And then the second experiment that we did here at Lawrence was taking these drug compounds and seeing if they can prevent the formation of hemozoin. Again, these drug compounds, we know that they work pretty well, but we don't necessarily know how they work. And the second experiment of seeing if they prevent the formation of hemozoin by causing a buildup of this oxidative stress in cells, this is a potential mechanism that these drugs could work by. So that was the purpose of the second experiment that we did. So in this first experiment where we tested these drug compounds on parasite viability, the first step was to harvest worms. So we had these worms that were infected with schistosomiasis, and the parasite was given time to develop and mature into these adult worms inside the mice. And then we would harvest the worms. So I'd like to specifically thank Stefan Debert, Judith Humphreys, Amanda Marsh, Ben Glazer, Marissa Zintel, and anyone else that I might have missed that helped us dissect these worms and harvest the mice. It's a very tedious process, and these mice are very tiny, about a centimeter or less in length, and very difficult to get out of these mice. So again, thanks to these people. Couldn't have done it without your help. And once we had the worms out, we would put them in this 96 well plate with cell culture, and we would incubate them for 24 hours to allow them to adjust. And again, the cell culture just had a lot of the same nutrients that the parasite would have in a human host. So the 24-hour incubation period here is just to allow the worms to adjust to their new environment. And then after they adjusted to that new environment, we added the drug compound and let the drug compound incubate in the cell culture plate for another 24 hours. And then at the end of this 24 hours, we used spectroscopy to examine which drugs worked. We essentially just looked at whether the worms were alive or dead. We did this using an MTT assay, which is essentially this compound that we put in with the worms and then take out after about 2 hours and then add in DMSO. And when we add in this DMSO, we either see a color change or we see no color change. And here we see if we see a color change and it's this dark purple color, that means that the parasite is alive. In other words, our drug didn't work because our drug is supposed to kill the parasite. And if we see a clear well, that means that the parasite is dead. So our drug did work pretty well. And specifically, MTT measures mitochondrial activity. So that's how we can tell which worms are alive and which ones aren't. We're assuming that if the mitochondria are working and producing this color change, that the parasite's alive. And if the mitochondria aren't working and they're not producing this color change, then the parasite must be dead. And here are the results of this assay. Up here on the left, you can see one of the plates that we use for spectroscopy. You can see that there's a lot of purple there. That means there's a lot of alive parasites, which means our drug didn't work at killing the parasites. Although we can see a few, like here in well 7, that are clear, which means the parasite was dead. And here we can see the graphs or the results from the assay. And in both cases, there's not really conclusive evidence for anything with these graphs. This is a difficult experiment to pull off when it comes to making sure the little worms are alive when we take them out of the mice and then keeping them alive for 24 hours before adding the drug compound. And there's just a lot of variables there that are difficult to balance, and only having a couple weeks to do this experiment, we didn't really get any conclusive results. But again, thanks to the Kaiser lab in Switzerland, we do have pretty conclusive evidence that these drugs do work at killing schistosomiasis. So we're going with those results from the Kaiser lab. And the second assay that we did was testing, do these drug compounds prevent the formation of hemozoin given free heme? So the first step in this experiment was to take a heme solution, which is just heme dissolved in an acidic solution, to match the pH in those digestive vacuoles inside the red blood cells. and we take the heme solution and we add a test compound to it. And again, this heme solution just contains 100% free heme. In order for heme to polymerize into hemozoin, the conditions need to be warmer and acidic. So to prevent that, we just store it in a fridge so we know that 100% of this compound is going to be free heme. And then we add the test drug compounds to it, and we incubate for 2 hours at 60 degrees Celsius. So this 2-hour incubation period allows all of this free heme to polymerize into hemozoin if the drug compound doesn't prevent it from doing so. So for example, in our negative controls where we didn't add any drug compound, we expect to see 100% hemozoin. So we would have no free heme in solution after these 2 hours at 60 degrees Celsius, where the heme has plenty of time to polymerize into hemozoin. After these 2 hours, we would add pyridine. And pyridine is this molecule we see here, just an aromatic ring with a nitrogen in it. And this nitrogen, as part of the pyridine ring here, can essentially just glob onto the iron core. This nitrogen carries a slightly negative charge, and the iron core has a slightly positive charge. And this nitrogen will coordinate to the iron core just like the other nitrogens in the ring of heme. And when the pyridine binds to the iron core, it causes a color change. It causes the color to go from clear to red. So again, we use spectroscopy to measure how red each well is and to see whether or not our drug compound worked. So in this instance, we know that our drug compound worked if we see a color change. So again, a color change means that the pyridine globbed onto the iron core of free heme. This can't happen to hemozoin, only free heme. So we know that there's free heme in the cell if the cell is red. And we know that there's not free heme in the well if the well is clear. So you can see here over to the right, we have a little bit of some red going on in some wells. And then here, like in row 1, we have essentially no free heme. So here are the results from our experiment that we did here. We can see a very clear S-shaped dose-dependent curve with our positive control of AMQ here. That's exactly what we would expect to see. As the concentration increases here on the x-axis, the absorbance also increases. And the absorbance here can also be viewed as free heme. Free heme and absorbance in this diagram are synonymous. So we can see as the concentration increases, so does the free heme, which is what we want. We want our drug to prevent the formation of hemozoin and cause this buildup of free heme to kill the parasite. However, we see our 3 test compounds here that are this orange, the gray, and the blue line. They have a little bit of interesting results here at low doses, but at high doses, they don't prevent the formation of any hemozoin. All formation of hemozoin essentially happens at these high doses. So we can essentially conclude that none of these 3 compounds prevent the formation of hemozoin, and they must be working by some other mechanism. And again, these 3 drug compounds, the Kaiser lab proved they are good at killing schistosomiasis. However, they just must be working by some other mechanism than the prevention of the formation of hemozoin. So future studies could examine different parasitic diseases or different drug compounds attacking this common pathway of the digestion of hemoglobin, where heme is formed and then the parasite takes this heme and makes hemozoin out of it. So again, this pathway is common to multiple parasitic diseases. Probably over 20 parasitic diseases at some point feed off the blood in humans. So any parasitic disease that feeds off the blood in humans has this pathway that's central to it. And a single drug that could prevent the formation of hemozoin among many different parasitic diseases could be very impactful to public health and to the world. I'll quickly go over the things that we would be looking for in this ideal drug that would treat a range of parasitic diseases. This drug would be orally administered because, again, these diseases exist in sub-Saharan Africa, a lot of developing countries that don't have the infrastructure to widely distribute drugs that have to be administered intravenously, intramuscularly. things like that. So an ideal drug would be cheap, safe, and effective, and also orally administered, um, so that it can be widely distributed to these areas that really need it the most. And again, a drug like this could have extreme lasting impact on the world. These diseases affect a lot of people. Again, over 200 million cases per year for each disease. So this is a lot of people that that their lives could be affected, their lives could be improved, and again, it would save and improve millions of lives around the globe. And finally, I'd like to thank these people, obviously Stefan Debert for being my research advisor. He's helped me a tremendous amount along the way, and I wouldn't be here if it wasn't for him. I'd also like to thank Judith Humphreys. We did a lot of our work in her lab and leaned on her knowledge. She's an expert in schistosomiasis, so she helped us out a ton there. Also, Eric Llewellyn in the biology stockroom and DMART in the chemistry stockroom, they helped a lot with my research, with getting me all the reagents that I need and things like that. Again, Amanda Marsh, Ben Glazer, and Marissa Sintel for helping us harvest the worms from these mice. That's a very tedious process. I'd like to thank Evan Oriel, Kelly Vensis, and Camille Kopp. They're my group members, and they helped me put together this presentation. Graham Sosma and Rachel Dowdy, who are our two professors for this class that taught us how to give great presentations. And finally, the Chandler Senior Experience that helped me by giving me all of the funding that I needed to do this research. And again, that's all I got. So thank you.
Very nice. Very nice.
Thanks.
Okay, so I think I'm running the floor here. So I guess Clayton will entertain any questions.
Absolutely.
I have a question. So is there any importance in the dimerization when talking about oxidative stress compared to the polymerization for the hemozoin formation? Like, would there be any point in trying to stop dimerization as opposed to stopping polymerization, or is that just not something that is important in oxidative stress?
It's more of a stopping the dimerization would be a lot more difficult than stopping the greater polymerization of hemozoin. And again, we don't have to completely prevent the formation of hemozoin. So just stopping the polymerization of those dimers is good enough. I'm sure it could be looked into, but it just doesn't really need to be, since stopping the polymerization of the dimers themselves is good enough. So if that answers your question.
Thanks.
So I'm used to thinking about MTT assays as with like a boatload of cancer cells, right? So thousands of cancer cells, you get a nice grade of, active to non-active. But you're sort of looking at one organism per well. And so we really would expect it to be on or off like the same deep purple or clear. That's kind of what I saw, but just numerically, is that what you saw, where all the numbers were sort of 0 or some big number like that?
There's actually a good amount of variation. If I go back to the picture of that plate, here we can see on the left here, I don't have this labeled, but rows or columns 1 through 6 are all male worms, and then columns 7 through 12 are female worms. And you can see the male worms are a lot deeper dark purple than the female worms. And I think that's because the male worms are just bigger, so they can convert more of this substance to a deeper dark purple compared to the females. So I think the deepness of the purple, I guess we could say, is more related to just how many cells and how many mitochondria are pumping out this purple substance.
And so you're not really putting any stock into the actual variation in absorbance. You're still kind of thinking of it in sort of Manichaean terms, up and down, or?
Yeah, it's more of a binary measure.
Yeah.
The the very light purple we could interpret as like 95% of the cells in these worms are dead, and there might be like this 5% that are still alive somehow, and maybe that's how we get a very light purple. But yeah, it's as far as analyzing the data, we're looking at it as a binary of, is this dark absorption or is this very light absorption?
Do you see the same in that other, the other assay that you used?
No, the other assay is more of a gradient.
Okay.
Because again, this one we can have a very light dark would, a very light red would mean that there's just a little bit of free heme in the well, but a really dark deep red would mean there's a lot of free heme in that well.
I have another question, if that's okay.
Yeah, go ahead, Evan.
So on, I think it's slide 23, where you saw at low concentrations of your hemozoin assay that you have like some higher absorbances. Do you have any like idea why that might be?
I really don't. I don't think it makes any sense because again, like it's more of like a strength in numbers game when it comes to the mechanism of these molecules. We think anyways, if they work in the similar way that chloroquine does. So we would think if there's a lot more of these drug molecules floating around that they would prevent more hemozoin and cause a buildup of more free heme. And since we observe that exact thing that we expect in the positive control, yeah, these low concentrations are likely due to some type of methodical error that we were making in the experiment, and likely don't have anything to do with the way the drug is actually working.
I have a question about the drug resistance timeline. You said that schistosomiasis already has sort of an established drug used for treating it that's working really well, and there isn't a lot of antibiotic resistance to it yet. And your drug is sort of like the future for when that happens. What's the timeline for antibiotic resistance? Is that like 100 years, 50 years? How soon are we going to need your novel drug compound?
Yeah, we don't really know when resistance is going to happen. We can look at other diseases to find kind of a ballpark. And the way that usually happens is we look at other drugs that have a similar mechanism. Like, for example, chloroquine prevents the formation of hemozoin. We could look at other drugs that also prevent the formation of hemozoin to find when resistance might occur. The problem with praziquantel is, again, that we don't really have a method of how the drug works that has really been nailed down. So we don't really know is the long and short of it. Yeah, the drug likely affects the nervous system, and nervous system affecting drugs generally don't have a lot of resistance against them, which is probably why schistosomiasis has this one drug in praziquantel that works for all cases of schistosomiasis, and it works really well and has for years and years. So again, it's likely that resistance isn't coming tomorrow, but at some point in the future, resistance is almost inevitable for all of these diseases.
And correct me if I'm wrong, but praziquantel is effective against the adults, but not the schistosomia form of the parasite, or is it vice versa?
Yeah, that's correct. Schistosomiasis is only effective against the adults because the adults are the only form of the parasite that has that complex nervous system that it can affect. Yeah.
So ideally, we'd like to develop a drug that's effective against both the schistosomia and the adult stage.
Yeah. Yep.
Cool.
Which these drugs that we've developed are in vitro, but not in the mouse model so far.
Is the drug something that people would take preventatively, or would we spread out as much of it as we can to as many people as possible to try and like wipe it out in people, or is is that a thing that we would do?
Yeah, so going back to the life cycle of these diseases, it could really work either way, because if any strand of this life cycle is broken, the life cycle can't continue. So if we just mass distribute it and it can only attack the adult worms in humans, but we can wipe it out completely in all humans, then we could essentially wipe out the disease, because we would have kind of a log jam at this Cercariae stage. And if no humans are infected, then we could essentially prevent, like, the parasite would just die off, because it needs the human host to survive. And similar in malaria. But yeah, the drug could be taken preventatively or to actually treat the disease. But again, in these countries, the numbers of infections are they can be interpreted in a number of ways, because a lot of these people can't go to the doctor to seek help. And a lot of people slip through the cracks just because of the infrastructure in the countries where the disease is most prevalent.
Thanks.
And of course, we could also treat the snails, but the hard part is making the pill small enough.
Yeah, and teaching the snails to swallow pills.
Exactly, it's really difficult. Okay, any final questions for Clayton? All right, great. Well, let's I guess unmute and thank him again for a great talk. Good job.
Thanks again, everyone, for attending my talk. Means a lot to have you all here.